So as I'm working on the area between art and science, my current project deals with portraiture of DNA and protein molecules. I'm interested in showing in my portraiture the fragility and complexity of these tiny molecules that are so sensitive and vulnerable. Thinking about the genre of portraiture, about what portrait really means, I noticed that very often when somebody is painting a portrait, there's a transition that occurs when the image goes from the real world into to the painting. There's some kind of abstraction that occurs, but at the same time, some of the fundamental features remain the same, and they are unchanged. And these features and these things that do not change are actually the connection between the painting and its concept. For example, here, the concept of this painting that I'm showing you now is the Queen of England. My portraits, their concept is in general science or DNA in particular. So I started working with Professor Brandman from Stanford University from the biology department. And his project, which fascinated me from the start, was a breakthrough in DNA studies, which he has done in a collaboration with Professor Frost from Utah University and Professor Weissman from UCSF. And this project brought me back to the laboratory where I wasn't for quite a few years. So all of a sudden, I came back to this space which was full of tension, where you see results of experiments and you anticipate for new results to come. You bring ideas, you analyze them. And all this dynamic really became very nostalgic for me, very beautiful. And on the other hand, I was an outsider. I was not now any voice in these experiments. So I took this standpoint of just looking at the visual side of these images. And suddenly I was interested not in what's happening in the results themselves, but how they look. So the first thing that I noticed when I entered his laboratory were these many sheets of what is called electrophoresis analysis. And electrophoresis is a very basic analysis of protein and large molecules that is so fundamental that most scientists don't really pay attention to it. And there are hundreds and maybe more images like this all over the lab. So I collected them and started to paint them in watercolor. And as I started to paint more and more in watercolor and in ink, I noticed that this transparency is actually almost made for watercolor, this type of work. I've done some of these works. And then simultaneously, I also started to work on video, that I've made animation from these still paintings. And as you can see, and once I finished the video, Professor Brandman composed the music for it. So I'd like you to hear this. If I can make it work here. So this was a very indirect way of looking at molecules. And the problem here is that I'm trying to give a visual language for something that is smaller than the wavelength of light. And therefore, you cannot really see it. So how do you do this? How do you go about seeing something that you cannot see? And that's what actually scientists have been doing throughout the years. And one of the most common ways to do this is through a microscopy, which is the most direct. But of course, you cannot really see in a microscope, not even with an electron microscope, the details of the actual atoms and molecules themselves. So what Professor Brandman was doing, he was looking at this type of aggregates. So these are groups of molecules, and he's trying to deduce from this structure what he can learn on the individual from them. And when I receive these images, they are not flat like what you see on the screen, but actually they do have a dimensionality. So when you look at them, you see a very deep abstract array like this. And I tried to transfer it into my art. And first I did these kind of drafts with the Perspect installations, and later decided to transfer them into fabrics. And these are layered fabrics where I try to convey this kind of depth and the layering that I get from the laboratory results. But as I'm looking at this type of work, this is something that actually I used to think about also when I was a scientist myself, that all the time you're looking at these samples, especially when you are a chemist, you're looking at these small samples, and you're always looking at them from above. You never really see what's in there. They are always much, much smaller than you, even when they are so much magnified, like you can see here in this image. In a way, you have this view like a creator, like you are the creator, which is something like this. Now, how much can you see from this image about the individual? Very hard to tell, but there are things that you can say from this collectiveness. So what I decided to do is to take the images and to magnify them, and I magnified them to a size that is larger than life, and created these installations from them together with videos, which you can see here. I've also done a different type of video, which this is one of the stills from it. Another way of looking that is more direct at these images is by what is called X-ray diffraction. And when you look at an X-ray pattern, the way you analyze images from them is you put the sample right here, okay? And then the result that you get is this kind of rings, okay? And these rings are sort of like the shadow of what the sample is. And by a lot of mathematics that we are not going to get into now, you deduce this type of structure, for example. That's the DNA structure, for example. So what happens here is that actually the final image that you see here, lost my arrow, but the final image that you see here on the right side is actually not the real picture, but what we deduced from its shadow. If we are taking an example for this, we can look that from this picture, if, for example, we could not see the people and could only see their shadows, think about what could we have actually learned about what these people look like. We could have a pretty good idea. We could deduce that they have a head and arms and legs, etc., but we could not really know what they look like. And that's the situation until today with the molecules and the atoms. We really cannot see them, although we have a pretty good idea of what their structure is. And with this in mind, that scientists are very aware of this. Every time that they are using these models, they are not using one model when they are looking at something because they know that no one model describes what they're seeing, but simultaneously they describe here, I just saw 3 examples of what DNA models look like. The one in the middle describes only the chemical bonds. The one on the right-hand side describes only the energy surfaces of these materials, and the one on the left approximates this kind of strips like in a ribbon, what is called a ribbon model. So what I did is I tried to draw my molecules by drawing simultaneously the different models for them. Like, for example, this is one of the models that I get from the laboratory, and then I'm doing this is watercolors where I do different kind of structures. The one on the right is from the molecular bonds, and the other one is the ribbon model. This is another example, and that's the third image from the same series. I try to place them one next to the other in this type of installations that I've shown in the past. But none of these is actually what it looks like. So what I did next is I started to draw them in layers. And this is from an image of one of the proteins called ribosome. And when I'm transferring it to my work, I try to show in the following installation that actually this is not what it looks like, but it's somewhere hazy in between this and another structure. So I'll show you this for a second. And this is what it looks like. So I just did a lot of these, I've looked at a lot of different imagery, and I'd like to finish my talk by showing you another clip of animation that I've made, which is using two types of other laboratory experiments. This one is mass spectrometry, where which is a way to identify proteins and DNA. And when you're looking at it, what I really loved about it is the results really have these wonderful points that reminded me of pointillism, in the history of art, which I thought was really just beautiful, and I started to work with it. I also used this tree diagram, which is the method of sequencing DNA, which I also thought is very beautiful with the wonderful colors and linearity. Also for this, and the end of this, video will be, as out of all these methods come out, there's the birth of the model that is coming out of this. Now, this model, as you will see, is not what protein actually looks like, not what DNA actually looks like, but it's sort of an avatar, something that is not real, something that is not there, but on the other hand, it's a pretty good signifier, and it's pretty useful for the entire fields of sciences. The music for this video was also composed by Professor Brandman. Thank you. That's it.